Hello there, welcome to DNA Edits. Today we will discuss about the terminal deoxynucleotide transferase. Terminal deoxynucleotide transferase. This is an unusual enzyme. Why I'm saying almost all the polymerases which are template dependent polymerases means to synthesize the new strand to add to the existing strand, they require the template. But when it comes to the terminal deoxynucleotide transferase, it does not require the template. So hence it is called as template independent polymerase. Template independent polymerase. Yes, you see, it possesses the unusual ability to incorporate nucleotides in a template independent manner using only single strand DNA as the nucleic acid substrate. Very clear that this enzyme can use the single strand DNA as a substrate to add or to catalyze the incorporation of nucleotides at 3' end. Let us see some of the important features of terminal deoxynucleotide transferase, because it is very important enzyme in the molecular biology and RNA technology and cell biology also. Right? Let us look into the unusual characters of this enzyme. Then we will see what are the applications of this enzyme. First, I just want to highlight how this TDT, terminal deoxynucleotide, is different from the other DNA polymerases and RNA polymerases. First, you see this is a DNA polymerase, where this DNA polymerase requires the primer. The length is between the 3 to 10 base pairs, and if you provide the DNA polymerase and the primer and template, that is in the double strand form, but I have taken only single strand, then what will happen? It will synthesize the new strand by using the primer as well as the template to synthesize the complementary strand is synthesized. So whatever nucleotides are incorporated at the 3' end of primer, they are complementary to the strand already there. They are not very different. So there are hence DNA polymerases and RNA polymerases, all of them are template dependent polymerases. So they'll synthesize the RNA or DNA only based on the template. The complementary strand is synthesized, but not very different from they don't synthesize the new DNA. They only synthesize the DNA to the existing DNA. But when it comes to the terminal deoxynucleotide transferase, you see, this is what is a strand, DNA strand, and you provided with the DNTPs and enzyme. Now the DNTPs or deoxynucleotides are incorporated at the 3' end of single strand DNA without need of template. You see, just they'll incorporate or polymerize these nucleotides at 3' end of single strand DNA without requirement of the template. Hence, the terminal deoxynucleotide transferase is called as a template independent polymerase. As on today, this is the only enzyme in the nature which can synthesize the template independent DNA. Remaining all polymerases requires the template. This is the key difference between the terminal deoxynucleotide transferase and the other DNA or RNA polymerases. You see, TDT, that is terminal deoxynucleotide transferase, requires the initiator complex for polymerization. So it requires the like a primer kind of structure with three nucleotides. If there are three nucleotides, then it starts polymerizing the nucleotides without template. So this condition is that the initiator or initiator complex or a primer, you say, it should have 5' phosphate and 3' OH. So the initiator complex, which is having the 5' phosphate and 3' OH, can only act as a initiator complex for TDT, terminal deoxynucleotide transferase. Here we have taken the trinucleotide complex, which is having the 5' phosphate and 3' OH. To that, if you provide terminal deoxynucleotide transferase and DNTPs, then it will synthesize the strand. It will incorporate the, or it catalyzes the incorporation of nucleotides at 3' end of the initiator complex. So you all know that DNA polymerases and RNA polymerases also require the primer, right? They catalyze the incorporation of nucleotide at 3' end, right? But their length varies from 3 to 10 base pairs. This is if the TDT is having 3 nucleotides, but at the same time condition is that 5' phosphate and 3' OH is very necessary for polymerization of the nucleotides at 3' end. So TDT, terminal deoxynucleotide transferase, requires the initiator complex whose length is 3 base pairs and 3 bases and consists of 5' phosphate and 3' OH. 5' phosphate and 3' OH only can, this complex only can initiate the polymerization of the nucleotides by using the terminal deoxynucleotide transferase. Yeah. So terminal deoxynucleotide transferase requires the metal ion as a cofactor. Not a single metal ion. It can use the 4 different metal ions. For example, you see, like any other polymerase, it uses the magnesium, it can use zinc, it can use the cobalt, it can use the manganese. It can use the manganese. So, like other polymerases, it requires the magnesium. In addition to the magnesium, it can use the zinc, cobalt, and magnesium. When it is using the terminal deoxynucleotide transferase, when it is using magnesium as a cofactor, there is a preference. There is a preference. It can add only purines. There is a priority to purines in the polymerization. You provided the 4 nucleotides, but still in the presence of magnesium, it will add guanine and adenine. Guanine and adenine. You see, all of them are guanine and adenine. Here I have, there is a C, but actually that is wrong. So, terminal deoxynucleotide transferase can give more preference to the purines over the pyrimidines in the presence of magnesium. Similar, at the same time, quite opposite to the presence of magnesium, when you provided cobalt as a cofactor, then pyrimidines are incorporated at the 3' end over the purines. So if you want to add only pyrimidines, then you conduct the reaction in the presence of cobalt, then the only cytosine and thymine are added at the 3' end of the initiator complex. So based on the metal, you can polymerize the pyrimidines or purines. That facility is there with the terminal deoxynucleotide transferase. Yeah. So this is a unusual and fantastic property of terminal deoxynucleotide transferase is that it can synthesize the oligonucleotide de novo. De novo synthesis means if you just provide the nucleotides, just you provide the nucleotides and enzyme, it can synthesize the nucleotide from two binucleotide or two nucleotide to 15 nucleotides in length. You see, just you are providing the nucleotides and TDD, it can synthesize the two nucleotide length, 2 base pairs or 15 base pairs. Maximum length is 15 base pairs. So you don't, for synthesis of 15 base pairs, there is no requirement of initiator complex. If you want more than the 15 base pairs length single strand DNA, then you have to add the initiator complex, whose length is 3 base pairs, consist of 5' phosphate and 3' OH. So this, you see, no other enzyme can do this one. De novo synthesis is not possible with any other polymerases, but only this polymerase can do it. Yeah. Like when you want to have a homopolymers at 3' end, and the requirement of the primer or initiator complex is different. For example, if you want to the polymer of T at 3' end, you have to take the initiator complex or primer length is more than 6 nucleotides. You see, the primer is having the 1, 2, 3, 4, 5, 6 nucleotides. More than 5. Generally, more than 5 is required to get the homopolymers. Just you provide the DTPs, DTTPs, and enzyme, and you take the 6 nucleotides length primer, then you get the polymers. Similarly, if you want to get the DAT, means poly A tails, then you have to use the more than 6 nucleotide primer. You see how many how many nucleotides are there? 1, 2, 3, 4, 5, 6, 7. Around 7 nucleotides, more than 7 or more than 6 nucleotides are required to synthesize the poly A tails. Poly A tails by using the terminal deoxynucleotide transferase. Yeah, so this is what is explained. Now we will we'll see with the text, right? Yeah, already we spoke about this one, and you see the TTD, terminal deoxynucleotide transferase, first time they isolated from the thymus gland. It's abundantly present in the thymus, but it is also present in the immature B cells and T cells. Why it is only present in the immature B cells and T cells? Because it is very, very important for the adaptive immunity or evolution of vertebrate immunity. Because of to evolve, to get the diversity in the immunity. So this enzyme is more useful. So enzyme immunity diversity is all depends on the TDT molecule, right? TDT, terminal deoxynucleotide, is a true monomeric protein. It's not a dimer. People thought it is a dimeric, but it's a monomeric, whose length is whose molecular weight is 58 kilodalton or 60. And this TDT, terminal deoxynucleotide transferase, is having splice variants. Depends on the species. In murine, it is having long and short, 2 splice variants, long and short. But when it comes to the human and bovine, it is having 3 splice variants. And one of the splice variant in the human shown that it consists of 3' to 5' exonuclease activity. So terminal deoxynucleotide transferase from human has shown that 3' to 5' exonuclease activity. Yeah, this is what you see. TDT plays a crucial role in the evolution and adaptation of the vertebrate immune system. This is very, very important enzyme for immune system. So because of terminal deoxynucleotide transferase, so you know how many types of antibodies are present in the human immune system? There are 10 to the power of 14 types of antibodies. And this is also one of the reasons for that diversity of the immune system. And when it comes to the T cell receptor, TCRs are almost like 10 to the power of 18. So in every antigen, almost every antigen present on the earth have an antibody in the immune system. That's all because of the terminal deoxynucleotide transferase. Yeah. The same thing I'm repeating. For polymerization of the nucleotides, it requires the initiator complex, at least 3 nucleotides, and it should consist of 5' phosphate, free phosphate, and 3' OH for extension. So initiator complex must have 3 nucleotides length and 5' phosphate, free phosphate, 3' OH. Yeah. Homopolymers all depends on the length of the primer. If there are more than 6 nucleotides, then you can get the poly T, poly A. If it is more than 5, you get the poly T homopolymers. So it can, since a de novo synthesis is possible with the terminal deoxynucleotide transferase, and it can use the 4 different metals, that is for catalysis of nucleotides. One is magnesium, zinc, manganese, and cobalt. The preference goes from magnesium to zinc, cobalt, and last one is a manganese. And I told you that in the presence of magnesium, purines are incorporated. In the presence of cobalt, pyrimidines are incorporated in the chain. Yeah, if you look into the applications, just I'm saying here for the sake of, since we talked about the enzymes, it is used in the synthesis of copolymers, means 2 nucleotides, like purines and pyrimidines, homopolymers at the 3' OH. And it is very, very useful molecule in the cDNA synthesis. After first strand synthesis, there is a need to second strand synthesis where TDT, terminal deoxynucleotide transferase, is very, very important. And the third most important one is that it is useful in the 3' end labeling. 3' end labeling. It does not have any substrate specificity. Because of that, highly radioactive elements, highly radioactive metal elements, nucleotides, and biotinylated nucleotides or fluorescinated, all of them are incorporated at 3' end. They're used as a probes for various purposes, like southern blot or colony hybridization or any whatever. Most of the hybridization techniques at 3' end, they use the terminal deoxynucleotide transferases. And terminal deoxynucleotide transferase is very, very important molecule in the identification of apoptotic cells, where tunnel assay is conducted. Tunnel assay. So the cell which is undergoing the apoptosis, if they incubated with the any fluorescent probe, and in the presence of terminal deoxynucleotide, this labeled probe labeled probes are incorporated into the DNA which is undergoing the division, that degradation, that indicates that the cell has initiated the apoptosis. You all know that during the apoptosis, cells DNA is condensed, then there is a fragmentation, kind of ladder kind of structure you can see in the cell which is undergoing the apoptosis. So to know the cell, whether is cell initiated the apoptosis or not, if you do the tunnel assay by using the terminal deoxynucleotide transferase, you can understand that whether it's undergoing or not. This is all about the terminal deoxynucleotide transferase. It is almost discovered in the 1960 by independent scientists at the same time, and it is very useful enzyme in the RDN technology and molecular biology. And but it did not, it structure, 3D structure was elucidated around 2000, right? So this is all about TDT. In the next lecture, we'll take up another enzyme. Thank you all for listening. If at all you like it, just subscribe for regular updates. Thank you all.